Hello everyone and welcome to my channel Be Yourself. This is Dr. Rajni Sharma and today I'm going to discuss the answer key as well as explanation for the part B of June 2015 CSIR-JRF NET Life Sciences questions. So let's start with the first question. The question is that a 1% solution of a sugar polymer is digested by an enzyme. The rate of monomer sugar liberated was determined to have a maximal initial velocity of 10 mg formed per minute. The turnover number will be. In my last video in my channel, I have discussed how to solve the questions related with the turnover number. And this is exactly similar to that. So I'm not going to discuss much about this. I'm just going for the formula related to the turnover number and how it will be solved. But the detail of the turnover number and the solution, the question related with that is in my channel. Be Yourself. You can get that the details of turnover number from there. So we know that the turnover number formula is K cat, which is the turnover number, is equals to V max by E total. E total. E total is the total number of total number of enzyme. Okay, so here the total number of enzyme we will take in the molecule in the number of as in relation with the number of moles, as there are 20 microgram of enzymes and having the molecular weight 200,000, then what will be the total number of enzyme? It will be 20 microgram divided by 200,000. Similarly, for the Vmax, here it is 10 milligram formed per minute and the molecular weight of sugar is 400. Another thing is that the unit for the measurement is here microgram and here it's a milligram. So we will change it accordingly. It's better to change milligram into microgram. So let's start the solution. The solution is that here we will express it in the mole. So it will be 10 by Vmax will be 10 by 400 divided by 20 microgram. So better I will change milligram into microgram. So it will be 10 to the power 10 to the power 3. 20 divided by 20,000. Okay, when we will solve this, you will get 2.5 into 10 to the power 5. So the answer will be number 4. Move to the next question. In an alpha helical peptide, the backbone hydrogen bond is between. There is no doubt the hydrogen bond is formed between the carboxyl, first carboxyl, with that of the N+4 or the fourth number NH group. So the answer will be number 3. Yes, about the stability. Following are the 3 single-stranded DNA sequences that form the secondary structures. So it doesn't matter whether about the structure. The only matter about the stability is that the GC content. The higher the GC content will be, the more the stable the DNA will be. So if you will see, the GC content in C is more as compared to A and then B. So the answer will be number 2. Move to the next question. Yes, the question is, which one of the following enzyme is not part of pyruvate dehydrogenase enzyme complex in glycolysis pathway? The first 3, pyruvate dehydrogenase, which is also designated as E1, dihydroxylipoyl transferase, designated as E2, and dihydroxylipoyl dehydrogenase as E3 are used during the glycolysis pathway, the dehydrogenation of pyruvate after the glycolysis, and then it will enter into the Krebs cycle. So the only enzyme which is not used is dihydroxylipoyl oxygenase. Next question is, yes, what phenotype would you predict for a mutant mouse lacking one of the gene required for the site-specific recombination in lymphocytes? What does the site-specific recombination do during immunity or during the development of or when a body gets infected with a foreign antigen? What happens? There is a site-specific recombination of antibiotic strain. We know that the strain has such some genes like gamma, alpha, and the most prone is the maximum of times we will get expression of immunoglobulin M. But if this site-specific recombination will not take place, then there will be neither formation of T cells or B cells. This is due to the lacking of the rearrangement of immunoglobulin. So it will reduce sorry, leads to the immunodeficient diseases. Next question is that the key determinant of the plane of cytokinesis in mammalian cell in the position of definitely is the central spindle, which is involved during the cytokinesis. It pull up the spindle fiber towards the pole and help in the equal distribution of chromosomes to its daughter cells. Next is that the beating of cilia is regulated by its nexin. I'm not discussing much in B sections. I am basically focused on the C section, which student basically feel difficult to solve. So in the question which I feel that need explanation, I am explaining only that question. Otherwise, I am skipping the question without wasting the time. Next question is cystic fibrosis transmembrane conductance regulator is known to control the transport of which ions? This cystic fibrosis is actually control the ABC transporter or the sodium, potassium, sodium and calcium pump. So it's the calcium ion. Next is the in bacteria heat shock response is temporarily primarily controlled by it's the sigma 32 because it controls some heat shocking proteins and it helps in the thermoregulation also. In type 2 splicing, what events will take place? In my video in the splicing of DNA, I already have discussed actually what happened. First of all, intron will bind with the 2' hydroxyl of the A adenine group and that it will form a lariat. After lariat formation, the hydroxyl of uracil will act on nearby phosphate group and it will be removed. Lariat will be removed and we will get the spliced RNA, and this is the type is group 2 splicing. So if you will move back, yes, it will be 2 prime. A free 2 prime hydroxyl of the internal adenosine makes a nucleophilic attack on the 5 prime P of the first base of introns, as you have seen that. So the answer will be 2 without any doubt. Next question is that given below are some of the statement about the prokaryotic and eukaryotic mobile genetic elements or transposomes. We know that in case of eukaryotes, it has next question is question number 31. Given below are some of the statements about the prokaryotic and eukaryotic mobile genetic elements or transposomes. So we know that in case of bacteria which don't have RNA basically have the DNA as the genetic material. And the next point is that in case of eukaryotes as RNA and DNA both are found and both can act as the transposomes. So the answer will be B and D, and as per this the answer will be number 4. Next question. Copying error occurring during replication are corrected by the proofreading activity of DNA polymerase that recognize the incorrect bases. What happened? Our DNA base gets replicated in the 5' to 3' direction, and also DNA polymerase also run in this pathway. So what happens when it is moving toward the 3', it's synthesizing 3', what happened? It in the same direction it will undergo the proofreading activity and it will again cut the nucleotide bases from 3' to 5' side, and then again it will add the 5' to 3' direction new nucleotides. So in this way it is double checked or cross checked by the DNA polymerase itself. So basically it has a 3' to 5' exonuclease activity, and likely the growing chain there will be at the 3' end of the growing chain, and remove it by not by as it were by and remove it by 3' to 5' exonuclease activity. So the answer will be number 3. Now next question. During each cycle of chain elongation of translation, how many conformational changes does the ribosomes undergo that are coupled to GTP hydrolysis? Actually, there are two times GTP hydrolysis takes place. Now look at this. What happened during elongation? First of all, what happened? tRNA along with TU and GTP bind with on the A side. After that, GTP is hydrolyzed into GTP, and then it will be removed out, and it is again used in the cycle after it is recycled with the help of GS, and where GTP is exchanged with the TU complex. Now, what will happen in the next step? Then the peptide transferase will transfer the formulated methionine to the second amino acid, and then it will the ribosome will move toward the 3' base site with the help of GTP elongation factor G, and in this way you can see that during elongation factor 2 times GTP is used and it is hydrolyzed to GTP. So the answer will be the 2. Yes, the next question is, collagen are the most abundant components of the extracellular matrix. In order to maintain normal physiological processes like wound healing, wound development, etc., which of the following types of enzymes is most important? This is basically a proteases, because now let's concentrate on the wound healing. What happened during that process when the surface gets wounded? Then there are certain factors, or we can say there are certain cascade mechanisms which carry out all the process during the wound healing. It can be intrinsic, extrinsic, or can be common in all the factors. If you will see that, suppose for example, when there is a surface wounding, then there will be the activation of factor 12, which is augment factor or serine protease. Again, factor 11 is also serine protease. So in this way we can see that the particularly this enzymes used during the process is the protease. Next question is, which of the following events never activate the G protein coupled receptor for sequestering calcium ion release? It's it's the activation of Frizzle by WNT, because if we will look at this, the interaction of binding to the sperm receptors, there is a release of calcium ion from the endoplasmic reticulum for its activation, as well as it also prevents the polyspermy. In DNA synthesis also, indirectly, the calcium ion regulates the cell cycles, and also the processes involved in the formation of certain proteins which are involved in the DNA replications. So the answer will be activation by of result by WNT. Yes, next question is, the main difference between normal and transformed cells it's without any doubt it will be immortality and contact inhibition. We all know that this is a property of a cancer cell. Next question is, when bacteria growing on the 20 degrees Celsius are warmed at 37 degrees Celsius, they are most likely to synthesize membrane lipid with more. It says long chain saturated fatty acids, because we know that if it will be saturated fatty acid, then this extra heat will be involved in to maintain the configuration of the membrane structures, and it will not disrupt the bacteria or kill it. But if it will be short chain or an unsaturated, it will have the lower temperature for its rupturing. So, or we can say that it will disrupt the fluidity of the membrane. As I have discussed this about the fluidity of membrane in my video in plasma membrane, you can get the link from the description if you have missed that. Now move to the next question. Hydra shows morpholactic regeneration and involve which one of the following signal transduction pathway in its axis formation? It doesn't need an explanation. Without any doubt, it needs the WNT beta catenin pathway. Next is the mammalian oocyte prior to sperm entry is arrested at what stage of cell cycle? It's basically metaphase of meiosis 2. It also doesn't need any explanations. It's just a theoretical portion. Sorry. In this video, I am basically focusing on the very few of the questions which really need explanation, because my main target is the part C questions. So, the pluripotency of inner cell mass in mammals is maintained by the course of three transcription factors, namely OCT4, SOX2, and nano-G. So the answer will be number 1. I am just skipping most of the questions. I only explaining the questions which I need that you need to explanation, because I'm going to focus much on the part C. Yes, here next question is, which one of the following statement about leafy, a regulatory gene in Rhabdopsis thaliana, is correct? It's definitely leafy, which is involved in the floral meristem identity, and from this concept also this also involved in the ABC modeling of flower formation, which I will explain in my further videos. Don't worry. Actually, here the question is little bit confusing, because here it's a leafy and and here also the leaf is present. So most of the students get confused that it may be involved in the leaf expansion, but it is the floral meristem identity. Next question is that the quantum yield of oxygen evolution during the photosynthesis drastically drops in far red light. This effect is known as red drop, and when we have certain exposure of red and blue light to the plant, then there will be the enhancement of the photosynthesis, and this is known as enhancement effect. Next question is, dark ground seedling display triple response when exposed to ethylene. Which one of the following is not a part of triple response? Actually, the triple response include reduced stem elongation, increased lateral growth or swelling, and abnormal and horizontal growth. So, as per this, the rapid unfolding and expansion of leaf is not the triple response. Next question, which one of the following compound is generally translocated in the phylum? It's definitely sucrose, which is synthesized during the photosynthesis also. Nitrogen gas is reduced to ammonia by the nitrogen fixation method. In order to execute the process, which one of the compound is usually required, and during that process ATP is required. Yes, the next question is, the S wave of normal human ECG originates due to yes, ECG graph, if you will remember, it's it's something like this. Yeah, there will be the isopotential line or isopotential or baseline. Then there will be the P curve followed by the Q, then R, S will be down, and then again with some curves there will be T. So it is P, Q, R, S, T. I haven't drawn it very nicely because it is a little bit difficult for me to write on my bamboo pad. So here the P denotes the development of action potential at SA node. This time atrial muscles are depolarized. Now here it is asking about the S, and QRS represent the repolarization of atrial muscles and depolarization of ventricular muscles. With particular on how this how this signal is denoted, PQ represents the contraction of atria, QR the spread of cardiac impulse from SA node to AV node, and RS the spread of impulse from AV node to Purkinje fiber. It also initiates the contraction of ventricles. Ventricular contraction continues till S to T, and it is followed by the ventricular relaxation. So, as per this, if you will see the number B, let depolarization of ventricular wall moving back toward the AV junctions. Next question is, which of the following skeletal muscles of human body contains a higher number of muscle fibers? Definitely the muscles of leg. Next question is BMR. BMR is is the basic basal metabolic rate, and it differs from men to female, and it is represented by weight to height ratio as per the age also. So if you will see, metabolic rate is very much lower in case of sleeping. Next question is, which of the following combination must be present in a steroid receptor that is located in the cytoplasm? Definitely, when the stereoreceptor is located in the cytoplasm, it will have to move towards the nucleus. So, it should have a analysis sequence, and such analysis sequence is localized in between the zinc finger motif. Yes. Next question is, segregation of allele can occur at anaphase 1 or at anaphase 2 of meiosis. With reference to this statement, which of the following organism is an ideal model system of identifying stages of allelic segregation in meiosis? Actually, Neurospora crassa has spore-like formation. So, if you will remember this structure, they have ascus formation. So, this is a little bit easier to analyze all these things in Neurospora crassa. So, the answer will be number 1. Yes, question number 51. The gene A, B, and C control three phenotypes which are sought independently. A plant with the genotype heterozygous A, B, and C is celled. What is the probability for progeny which shows the dominant phenotype for at least one of the phenotype? Here it's highlighted as at least one of the phenotype, and we know that if it is the three allele is here, so we will have the total number of 64 progeny, and there will be only one chances where all the locus will be in the recessive one. So the answer will be 1 by 64. Hybrid dysgenesis in Drosophila is caused by P elements. Which one of the following crosses between different cytotypes which lead to dysgenesis? Actually, dysgenesis is caused due to the absence of P element, and this P element is only possible or present in the female Drosophila. It will be transmitted with the help of or transferred to the progeny with the help of female Drosophila. So as per this, if there will be M type don't have the P element and P type have the P element, so if the M type which is the female one, and it don't have P element, so the one which will have to pass the P element. I mean to say is that if female don't have the P element, it has the M cytotypes, then the P element will not be passed to the progeny. But here the P element is present in case of the male one. So this dysgenesis will cause due to the mating between the M cytotype and P cytotype, which is male. If it will be P cytotype, then this condition will not exist. Next question is that in an environment, clones of plant is grown in a field. The plant were observed to be the different height when a graph was plotted for the frequency of plant y-axis against different heights. A bell-shaped curve was obtained. From the above, it can be concluded that observed variation in height is definitely it's due to the environmental effect. Which one of the following virus cause acute gastrointestinal illness due to contamination drinking water? Norovirus is the virus which transmit through water, food, and air also. The phylum in which the animal are bilateral symmetrical in the larval stage and radially symmetrical in the adult stage. And it is Echinodermata. I'm skipping most of the questions without explanation because I have told that I will focus on the section C. And here too, which of the following fungal groups has septate hyphae and reproduces asexually, budding conidia and fragments? And it is the Basidiomycota. Yes, ribose. Most common used Molecular tools for phylogenetic analysis involve sequencing of ribosomal RNA because it is very much specific 16S RNA sequencing we do. The dynamic of the any populations within the meta population differs from the normal population is that, let's see, I better skip most of the questions because it no need any explanation, it's just a theoretical portion, because meta population is controlled basically by immigration and migration rates which are significantly higher. If it would be low, then we can neglect it, so it should be high. Next question is, which of the following is not simulparous? So it's actually a reproductive strategy. There are two types of reproductive strategy. One is the simulparity and another is the heteropaterity. The species is considered simulparous if it is characterized by a single reproductive episode before death, and having the multiple is termed as heteropatry. On this basis, we know that bamboo also have single lifespan and it only reproduce only one time, so it will not be included in this. The only thing is which has mayfly also has short life, it die after one reproduction. So only is that Dracena. The general relationship between the generation type and population's growth rate is described by the equation. The equation is 1 ln r is equals to ln a minus b log t. Next question, number 61. Which of the following is likely to contribute to the stability of the ecosystem? If there will be increase in any carnivorous or only herbivorous, then it will not contribute the stability. Rather, it will focus on only one food chain. But if there will be more omnivorous, then it will lead to the balance between the carnivorous as well as herbivorous. So the answer will be more omnivorous. Next question is, in eusocial insect, males develop from unfertilized eggs while female develops from fertilized eggs. The ultimate consequence of this difference is that what happens if there are two eggs and one is fertilized and other is not fertilized, then what will happen? It will develop into female and other will develop into male. So definitely it may be possible that their offspring can be male or female. When it will lack egg, then it may be possible that this egg may be unfertilized and may develop into male. So it is known that the female which developed from such mechanism are more closely related with her sister than her own offspring. So the answer will be number 2. The extraordinary sensory ability that elephant possesses is it's due to its emission and detection of ultra low frequency sounds. Yes, according to which evolutionary theory there is a long periods without significant evolutionary changes interrupted by episode of rapid evolution. It is a punctuated equilibrium. The name itself suggests that it means some hindrance is there, but it leads to the equilibrium after interruption by a short episodes. It gets some full stop in their pathway. Next is that the mean and standard deviation of body size in a Drosophila population are 8.5 and 2.2 mm respectively. Under natural selections over many generations, the mu and or the mean and standard deviation of the body size changes to 8.5 and 0.8 mm respectively. Here you can see that mean is same, so there is a stabilization during the natural selection. So definitely the answer will be stabilization. Next question is, the researchers would like to monitor changes in the level of serum protein for which an antibody is available. Which one of the following method would be best suited for the purpose? It's definitely enzyme-linked immunosorbent, which is basically based on the antibody analysis. It can be sandwich, it can be direct or indirect ELISA. Next question. Which one of the following method listed below is the most sensitive label-free quantification method for protein? Actually, level free quantification method in infrared, also in Raman, also in C3, we have to take or have to level the protein. But in UV spectroscopy, you need not to level any probe to the protein for the identification. What will happen if DNA is labeled by Nick translations while doing footprinting? If it is labeled by the Nick translation, then the linear order of the fragment from 5' to 3' end of the DNA cannot be arranged. Next question is, yes, the protein has the one tryptophan and one tyrosine in its sequence. Assume molar extinction coefficient at 280 nanometer of the tryptophan and tyrosine is 3000 and 1500 per molar per centimeter respectively. What would be the molar concentration of that protein if the absorption at 280 nanometer is 0.90? It's a very simple question, but you will feel complicated because there are two values of efficient its extinction coefficient is given, one for the tryptophan and other is for tyrosine. In a protein, there have the maximum of absorption is due to the tryptophan and tyrosine, and both extinction coefficient is given here, and the formula of absorption is epsilon Cl. L is the path or the path length of the cuvette or the path length of the light which travels across the cuvette. It is standardized as 1 centimeter. We have to find out the concentration, and epsilon is given. Now, here the trick is that as there are two amino acids, so to calculate the extinction coefficient of tryptophan and tyrosines in the same solution or the same sample, we have what will be the absorption. It is due to because of the both presence of tryptophan and tyrosine. So we will add the value of extinction coefficient of tryptophan as well as tyrosine for this, which is 3000 plus 1500 is equals to 4500. Now absorbance is given 0.9, so now we can calculate the concentration by putting all the given value at 0.9 by 4500. When you will calculate this, you will have 0.2 millimolar. Now it was simple. Next question is, two groups controlled and treated are to be compared to test the effect of a treatment. Since individual variability is high in both groups, the appropriate statistical test is used to. Here you can confuse with the ANOVA because in this ANOVA also we have the same strategy. We apply the control and treated one, but the another thing is here is that there is the individual variability is high. If the individual variability is high, on that case we use the Mann-Whitney U test. So this all were in this explanation on part B. In my next video, I'm going to explain the part C section of June 2015 CSIR-NET. So don't forget to subscribe and share my channel. Till then, goodbye and have a nice day.